Welcome to this series of webinars on rabies presented by Defra today. My name is Dr. Helen Roberts and I am the Senior Policy Advisor responsible for rabies in the Exotic Disease Control team in Defra, and I'm very pleased to be introducing you today to several people, experts in their field, who will be explaining this disease to you. So, as a background, just to talk about our current threat level from GB's point of view, we have been free from terrestrial rabies since the 1920s, and the only cases recently have been in animals associated with travel and in non-compliant imports of those animals. We do occasionally get a human case where they have also been associated with travel to a rabies endemic country, but we have not had any human cases associated with indigenous rabies since the beginning of last century. Our risk management, because we have a very strict policy around rabies, is based on restricting the movement, or specifically the import, of pets, commercial dogs, cats and ferrets, and other captive animals which are considered reservoir hosts, through applying a series of measures which could include quarantine, vaccination, blood testing, and waiting for clinical signs to appear. We harmonised with the EU on our pet travel scheme back in 2012, and in order to do that we undertook a quantitative risk assessment which was tendered out to some other experts, and this concluded that the risk level was very low if we were to align with the EU rules. So, to put it more exactly, the results of the quantitative risk assessment assumed a 100% compliance with all our regulations, and it suggested that under the EU Pet Movements Protocol, or EU PMP, the annual risk of rabies introduction from non-UK cats and dogs coming into the country would increase from 7.79 times 10 to the 5 to 4.79 times 10 to the -3, and that equates as being one rabies infected animal entering the country every 13,000 years, changing to one coming into the country every 211 years. And that is still, despite it sounds like there's a big change in that risk level, it's still only a very low risk when you compare it to other threats to animal and public health. Then, as pet imports increased again when Romania and Bulgaria were given free movement around the EU, we looked at the numbers of animals and how that changed the risk level. And although the risk increased again, it still remained very low to low. And that's because of the improved rabies control situation across Europe, particularly across the EU, where widespread wildlife vaccination has been occurring now for a couple of decades, and that's brought the numbers down significantly. However, we are now in a place of crisis in Eastern Europe, and we are therefore helping Ukrainian people who are fleeing the crisis and want to bring with them their pets. And to do this, we've put in place certain measures to reduce the burden on the owner of what they would normally have to do if they were bringing these pet animals into the UK. It's risk-based and will still require quarantine or isolation, proof of vaccination and a wait period, but will depend upon the risk level of each of those animals. Some of the lower risk animals may have been moved to home isolation, but we recognise that there's still a risk for an animal that could be incubating rabies prior to vaccination and just after vaccination, and could move before clinical signs become apparent. Now, this is likely to be a very rare event, even if it does happen. We're closely monitoring the same level of movements and types of movements across Europe. And if we do get a case of rabies in a pet belonging to a person from Ukraine, we need to be prepared to act. And part of that preparedness is alongside exercising our capability, which we've done recently, but we've also developed this series of webinars to help people understand the disease and the risk and the actions which can therefore be taken. Just to explain a little bit more about the risk of these pets coming into the country, This is a map of all the cases that have occurred between 2019 and 2022 in cats, dogs, wildlife and domestic animals. This does not include the numbers of cases of European bat lyssavirus, which we do have in the UK. Again, it's very rare, but it does occur sometimes in some of our Daubenton's and Serotine bat populations, and those numbers are not included in the classical terrestrial rabies cases that you see here. But as you can see from this map, most of the west of Europe is free. You have the occasional cases that turn up in imported animals, and usually, as I say, illegally imported animals. But there's a large number of cases in Ukraine, and that's the area that we're most concerned about, not just Ukraine itself, but also the potential movement of wildlife with rabies over the border into the other countries around them, Poland, Belarus, Moldova, Romania, Bulgaria, etc., and Hungary. So we are concerned about the potential for rabies to spread in these areas. And usually, because of the rate of reporting in Ukraine, there are rules around the movement of pets which are more stringent than those for other listed third countries. And as you see here, the rules that can be applied are based on vaccination primarily, and then a wait period before the animals can move. And because this is Ukraine is considered an unlisted third country, then a blood test has to also be undertaken, and it must be positive at a level of more than 0.5 international units. And that's for rabies antibodies, which is a way of proving that the animal has been vaccinated correctly. And then a 3-month wait is undertaken, during which time, if the animal was already incubating rabies at the time of vaccination or before the vaccine becomes effective, then in the vast majority of cases, the disease will have manifested already. And it's that period of time which is potentially a very long, but very rarely very long period. So we do know that sometimes there have been several weeks, if not a small number of months, before the animal has shown the clinical signs associated with rabies infection. But the majority will show signs within a couple of weeks. So by undertaking this risk-based approach to how these animals are arriving in the UK, then we are able to manage the risk from the Ukrainian pets. So the following few seminars that you will see will explain more about the disease itself, about the epidemiology, and also about the public health risks associated with it, and what we're doing to diagnose, test and manage the cases. Thank you very much.
Hello, my name's Bethan Donovan and I head up pet travel in Defra. Defra have taken a number of measures to support Ukrainians fleeing the conflict with their pets. The usual health requirements for someone bringing a pet from an unlisted country such as Ukraine are for the pet to be microchipped, vaccinated for rabies, have a follow-up blood test 30 days after vaccination to be carried out by an approved laboratory, followed by a 3-month wait from the date the blood test was taken. Dogs will also need a tapeworm treatment. We have not relaxed these requirements. However, we do not require pets accompanying Ukrainians fleeing the conflict to have gone through all of these preparations before travel. They can instead enter quarantine on arrival in the UK so that these preparations can be carried out here in a safe environment. Defra has streamlined the application process for people fleeing Ukraine with their pets. A dedicated helpline has been set up, and additional staff have been trained to manage calls from Ukrainians and their sponsors. There is a shortened application form for pet owners to complete. APHA helpline are supporting people with completing the forms where necessary. The application forms are available in English and Ukrainian. Ukrainian-speaking members of staff are on call to support the APHA help desk. The gov.uk page has been updated with advice on how to bring your pet to the UK if you are fleeing the conflict. The page is available in English, Ukrainian and Russian. We have produced flyers in English, Ukrainian and Russian for the visa processing centres and transporters to share with Ukrainians who do not have access to the internet. This sets out what they need to do to bring their pets to the UK. Once here, APHA will arrange a quarantine space and government will cover related costs. We have increased official quarantine capacity and are working with boarding kennels and rescue and rehoming centres across the country to ensure isolation capacity is available. We have doubled the number of quarantine spaces. We have increased APHA resources at the border to support the transport of pets to quarantine. Government will cover the cost of quarantine, isolation and all necessary veterinary treatments. This includes arranging and paying for pets to be collected at the border and transported safely to quarantine. Emergency legislation has been passed to permit the use of the ELISA test to verify successful rabies vaccination, which will reduce the amount of time a pet needs to be in quarantine. Once a pet has passed a blood test and left Ukraine more than 30 days ago, it may be eligible for home isolation or will be moved to boarding kennels for up to 3 months.
Hello, I am Catherine Grace. I'm a veterinary advisor for epidemiology and risk here at the Animal and Plant Health Agency, and I'll be discussing the transmission of rabies and options for disease control. So rabies is a Lissa virus. It used to be referred to as classical rabies, but is now just simply rabies virus. It can infect and replicate in a wide variety of mammalian hosts. It's most commonly, the urban cycle is most commonly associated with the dog. The virus tends to circulate within one species, but you can get cross-species transmission as well. So, as I say, most commonly associated with transmission within the canine population, but then you can get transmission from dogs to other species, such as cats, humans, livestock species, which tend to be a dead-end host, and wildlife species such as foxes or raccoon dogs. And transmission can occur within wildlife species. My colleague Graham will be discussing the sylvatic cycle within wildlife. I have included an image of bats because bats are really important in the transmission of Lissa virus and rabies virus, but I will only be talking about the terrestrial species, in particular dogs, in this section. So before thinking about the pathways of transmission, I thought it would be useful just to go back and review the pathogenesis. So the virus is inoculated into muscle tissue or directly into nerve tissue via a bite or licking of broken skin. There'll be local replication within the muscle tissue, and then there's retrograde axoplasmic flow of the virus along the peripheral nerves, so ascending up to the central nervous system. And then once it reaches the CNS, there'll be rapid viral amplification, and it will travel into the brain, with the brainstem being the first part of the brain to be affected. And the clinical signs, which Luke Gamble's going to discuss in another section, are the result of disruption and dysfunction of the nerve cells within the brain and the encephalitis that's caused by the virus. So it disseminates around the brain, and then it travels to peripheral sites, in particular the salivary gland, which is obviously where it's then primed for further onward transmission to other animals via biting, but it can also go to the ocular tissue and to the other organs. So then, so obviously, the transmission routes are quite simple after that. The most typical route for transmission of rabies is via bites or scratches or entry across damaged skin following licking. Atypical routes are described, so you can have entry of the virus via oral or ocular mucosa. There are reports of patients who have developed rabies infection having had an organ transplant from somebody with rabies. It is possible that infection could follow ingestion, but it's thought that would only be possible in the instance where the gastrointestinal tract mucosa has damage to it, which facilitates the transmission across that broken barrier. And also important to note as well that the virus does not survive well in cadavers or carcasses, and so that's considered to be a sort of unlikely route of transmission. Aerosol has also been reported, so inhalation has occurred. There are instances in the bat cave where there's a huge population of bats, and people working there were thought to have developed infection after inhalation. There are also reports from a laboratory setting, but these are very, very infrequent and very unlikely, and absolutely biting, scratching and licking of broken skin are the most typical route of transmission. It's important to note that the virus does not survive well sort of out in the environment. So once desiccated, it will no longer be infectious. Heat and light render it non-infectious. So contamination of fomites and the environment is not a concern in the epidemiology of rabies, as it would be in other transmissible diseases that we think about. We move on to think about the disease dynamics. The period in between exposure, so that biting or licking or scratching, and the development of clinical signs is very, very variable. The incubation period can be a few days, it can be several years. Usually it will fall within 20 to 90 days, but it can vary widely. And the reason for this variation depends on a number of factors. So that can be where the bite occurred. So if it was a very shallow bite in the muscle, it will take time to replicate and for it to travel from that location of the bite to the nervous tissue. And so the further away it is from the nerves, then the longer the incubation period. But once inside the nerves, it ascends at a rate of about 50 to 100 millimeters per day. But obviously, if it's inoculated straight into the nerve, then the incubation period will be shorter than if it's inoculated into muscle tissue on the periphery. The infectious dose is very important. So a high infectious dose is likely to result in a shorter incubation period, and a low infectious dose would be a longer incubation period. The immune status of the animal is relevant. So has that animal been previously vaccinated, which might slow down the incubation period? Does it have any concurrent diseases which might speed up the incubation period? And also the species or strain of virus that is inoculated is really important. So there is some host adaptation between strains, and when inoculated into a different species, say from dog to cat or dog to cattle, for instance, then you might see that the incubation period is variable between those two species as they respond differently to the strain. The infectious period is the period of time when the animal can transmit virus to others. And again, that is very variable, and it's short, usually somewhere between 4 and 7 days. And once the clinical signs have become apparent, that clinical phase where the animal is displaying clinical signs is short and is usually 7 days or so. So that short incubation period corresponds to rabies having a very low R number. So R0 is the average number of secondary cases that will occur from a primary case in a totally susceptible population. And it's been estimated by Hampson et al. that figure will normally be somewhere between 1.2 and 2. It's important to note that that is an average, and so you can get some animals which are super spreaders and will infect more than 2, but the average lies somewhere between 1.2 and 2. So then if we think about what are the options for controlling or preventing disease in animals, well, culling, isolation and tethering will prevent transmission from an infected animal to a susceptible animal, and will have an impact on the overall prevalence, but is unlikely to be a sufficient method in its own to eradicate the disease, partly because the infectious period occurs before clinical signs are apparent, so it's difficult to identify the animals which are incubating but don't have clinical signs. So that means that control can be tricky. Intuitively, I mean, reducing the population size seems like it should be a good option, that transmission is likely to be greater where there's a denser population. But actually, when you reduce the population size, so the culling of sort of all animals or some dogs in an area to reduce the population size or density, again, is not effective on its own. And in some situations, you might create an area where there are fewer dogs, and you create a niche which is then attractive for other dogs to move in. So you might encourage greater movement and spreading of the disease. Reducing the number of strays and roaming dogs, managing the population, again, it should reduce the opportunities for exposure between infected and susceptible animals. But ultimately, the number one measure for preventing or controlling rabies is vaccination. So there are lots of efficacious vaccines available. Because of that low R0 number, correspondingly the herd immunity threshold, so the proportion of the population that needs to be vaccinated in order to get R below 1 and to stop the transmission of the disease, is low. So if we use that R0 of 2 to work out the herd immunity threshold, it gives a value of 50%, which is relatively low. However, in reality, the coverage needs to be higher, and it needs to be frequent, as the population is turning over, that there'll be puppies entering the population which will be susceptible, and that will happen frequently. And also there'll be some dogs within that average who will be infecting more than 2. And so in reality, you need to have a higher coverage, and it's normally, so the aim is often about 70% for herd immunity. So that was all I was going to cover in my section today, and here are my references. I haven't discussed clinical signs in my section, and that's partly because it is not a disease which I can admit to having seen, and rather than me sort of reading the textbooks and reading papers, we thought it would be more useful to have insight from someone who has worked extensively on rabies and has seen it in real life. So we're very lucky to have been joined by Luke Gamble, who's the Chief Executive of Mission Rabies, and has very kindly joined us today to give an overview of what to expect from clinical signs of dogs infected with rabies. So he will be leading the next section. So thank you very much to Luke and the team at Mission Rabies. And that's it from me. Thank you.
Hi everyone, Luke Gamble here from Mission Rabies. I've been asked to talk to you about the clinical signs of canine rabies. So let's get cracking with that. What is rabies? Well, I think the dry form of saying it is that it's a neurotropic lyssavirus that tracks up peripheral nerves and causes a fatal encephalitis. But the key sort of points are that it's the world's deadliest zoonotic disease. It affects all warm-blooded mammals. Over 99% of all human cases are from dog bites and saliva, and there's virtually no treatment once the clinical signs manifest. From a welfare perspective, it's the reason why tens of thousands of dogs are killed all over the world from fear of this horrible disease, and pertinent to human context, a child dies of canine transmitted rabies about every 9 minutes. So it's a horrifying disease, and it's a neglected zoonotic tropical disease. Rabies is endemic in about over 150 countries worldwide. So you can see here on this WHO map, the areas in blazing red are high risk and the areas in orange are medium risk. So dogs coming in potentially from the Ukraine, you can see there the sort of medium risk area. So it's something we need to be thinking about and be aware of. And what are you looking for? Well, as Catherine outlined in her section, the incubation period varies quite a large amount with rabid dogs. Typically, we see rabid dogs show signs about 2 to 3 weeks after having been bitten by another rabid dog, if they're unvaccinated, of course, but it can be 80 plus days. The first symptoms are really non-specific. The dogs are really out of character, they're out of sorts. So you might get the lethargy, fever, anorexia, they're generally just not themselves. And that is really the alarm bell, because it's very hard to jump to thinking, that dog is rabid. This little puppy on the screen here that you can see, this puppy was actually brought to our clinic in India, in Goa, and it was out of sorts, not itself, and it nipped a few people. It had rabies, and when we obviously the clinical signs progressed fairly rapidly, and we put it down and we tested it, and it was rabies positive, and it had bitten 14 people, come into contact with 14 people before we got it. And many of those people would have just thought it was a bit of a naughty puppy. And that kind of emphasizes the danger that we're dealing with here. Typically, after the first symptoms sort of have kind of started to manifest, within days, they will progress quite seriously. So you see cerebral dysfunction, cranial nerve dysfunction, you get ataxia, weakness, you can get the paralysis, seizures, hypersalivation, difficulty swallowing, all that sort of stuff. And there isn't a one list of symptoms fits all, so you can get a variety of clinical signs. The aggression is a factor, and that really just relates to a dog being, starting to suffer horribly. If they've got a horrible pyrexia, horrible temperature, and they're not really quite sure what's going on, they're disorientated themselves. They do act aggressively, and they'll bite, and typically they just won't let go. I'll show you a little video in a minute of some of the rabid dogs we've picked up from our clinics in India. Death follows about 5 days after showing clinical signs, to give you an idea of what to expect. So let's have a look at what rabies looks like.
Yeah, okay.
So you can see there, they are quite sort of non-specific in what they do, and they are hyperstimulated, I think is probably one way of describing it. And ataxia, you might think maybe it's got distemper, maybe it's got a meningitis and encephalitis, but invariably what will happen is the dog will rapidly deteriorate. And as I say, after about 5 days of showing this kind of signs, they sadly die. And poor creatures, they really suffer with it. So in total relevance to the situation, what do you do if you suspect a rabid dog? Isolate the dog, call the Defra helpline, and then crack on with tracking details of anyone who's come into contact with the suspect animal to initiate post-exposure prophylaxis. I'm sorry for these fairly grim pictures of people. These are kids that were bitten by rabid dogs in India that our teams identified, and then they all survived, and we made sure they're okay. But the key thing, often people will just brush it off. They might not worry. They'll just, wash their hands and be done with it, or wash the wound, not for long enough, and then they are seriously at risk. So do crack on, track details of anyone who's come into contact with a suspect animal. Make sure the animal's isolated, and call the Defra helpline. Thanks very much.
Hello, I'm Graham Smith at the National Wildlife Management Centre, APHA, and I'd like to tell you a little bit about rabies in wildlife, specifically in bats and foxes. And you can see here at the bottom left some bat sampling we occasionally do, taking a mouth swab from a bat, and at the bottom right an image of a fox taking baits. Now, rabies is the disease, but it's actually caused by any of a number of lyssaviruses, and we're finding new ones all the time. Lyssaviruses tend to circulate in just one or two host species, but can spill over to give disease, resulting in death in most other mammal species. So in each area of the world, you tend to only have one or two host species primarily responsible for its spread. Now, classical rabies, or RABV, as it's formally called, is the one we're most familiar with and is the most common, occurring in dogs and wild carnivores. And this is the virus that causes most human deaths. Now, back in the 1880s, when Pasteur developed the rabies vaccine, there was only one form of rabies known. But genetic typing has now shown us a much wider variety of lyssaviruses. You can see here on the right the full list of them as of last year. Now, the majority of these circulate primarily in bat species, but of course they can spill over into humans and other animals. If we look first of all at rabies in bats, we can see across the Americas, we only have classical rabies, RABV, present. And this occurs in terrestrial animals, such as the skunk or the coyote, and also in bats, so some insectivorous bats and the vampire bats in Southern America. So the majority of lyssaviruses occur throughout Africa and Asia, and this is probably where they evolved and spread out. If we look to Europe, we can see a number of lyssaviruses here, but there's really only two that we're particularly concerned about, which is European bat lyssavirus 1 and European bat lyssavirus 2. So if we come a bit closer and have a look at rabies in bats in Great Britain, we can see the green dots in the bottom southwest of the country are EBLV1, and these occur primarily in the serotine bat. And the serotine bat is restricted to southern England and Wales. So it's not unusual those diseases that we found aren't widespread. EBLV2 occurs in the Deventons bat, and the Deventons bat occurs throughout Britain, and thus it's not that surprising that we see the occasional cases across Britain as well. We also have one unconfirmed case in red there in a pipistrelle. We knew it was rabies, but couldn't determine which one it was. Now, rabies in bats is still quite unusual and rare, and not a general risk to members of the public. But if you do come across grounded bats, it would be good to contact the Bat Conservation Trust. Grounded bats commonly happen in summer with juvenile bats, or when bats are a bit stressed or dehydrated. So they usually just need a little bit of care. Now, classical rabies in foxes was widespread in Europe during the last century, but has not occurred in Britain since the 1920s, when it was imported with people returning with their dogs after the First World War. Now, classical rabies spread in foxes throughout the latter half of last century, but has also affected raccoon dogs, cats, and other wildlife and livestock. Now, the typical incubation period is about 1 month before the animal becomes rabid. And during this period, you can't really detect the disease. There are no signs or symptoms. Rabid foxes often show this aggressive, furious, biting behavior that we associate with rabies, but can also show the paralytic, dumb form of rabies. And this is where they have progressive paralysis, usually from the back heading towards the forelimbs. In either case, the animal will die within about 5 days. Now, rabid foxes tend to be more active during the day, but don't usually wander long distances as rabid dogs might do. So the rate of spread in foxes would be relatively slow. Rabies is a notifiable disease, so any suspicion that a wild or domestic animal is rabid needs to be reported to APHA. However, other diseases can also cause strange behaviour, including making foxes bite people. Now, in urban areas, many foxes occur. They have little fear of people, so this is not a sign it has disease. Rabies control is the most successful wildlife disease control using vaccines ever. The oral vaccine was developed in the 1970s and 80s, and at this time fox rabies was in Northern Europe and a constant concern about it turning up in Britain, with people taking pets in from the continent. And we can see here a poster, La Rage, which is rabies in French, aimed at people coming across the channel during that time. Oral vaccines were distributed across Europe where wildlife rabies was occurring, primarily through aircraft distribution, but also by hand in urban areas. And we can see here an example rabies bait, about the size of a matchbox, with an aluminium capsule in it, which contains the vaccine. Now, when rabies was widespread at the beginning of the vaccination campaign in the 80s, you can see here it was spread up to northern France, across all of Central and Eastern Europe. It didn't actually take too long before this was pushed back with the vaccination campaign. And if we jump forward 10 years, we can see here at the turn of the millennium that most of Western Europe is free of rabies, with just a few foci left, which were quickly wiped out. And rabies, although it was still fairly common in Eastern Central Europe, if we jump forward to today, we can see the success of those continued oral vaccine campaigns across Eastern Europe. However, there remains a large focus of rabies in wildlife and dogs in Ukraine and Russia, and some countries such as Belarus, who haven't been reporting, we're not sure what's happening. But primarily across the EU countries, rabies has been removed in wildlife. Now, the UK has had a contingency plan to deal with a fox rabies outbreak since the 1980s. Any case would be imported through the movement of dogs or cats, or possibly other mammals, which is why such animals need to be vaccinated prior to arrival. But if an outbreak did occur, this would be a focal outbreak with very limited geographical extent. So, given the success of rabies elimination in Europe, we can be very confident that oral fox vaccination would also eliminate rabies here in Britain. Now, to declare a country free of wildlife rabies takes 24 months with no cases. So the vaccination would have to continue for at least a couple of years. And this would involve distributing the oral vaccine at least twice a year. If you'd like any further information on it, you can see the Rabies Bulletin Europe and the link here to the Bat Conservation Trust. Thank you.
So now we're going to look at reporting suspicion of rabies. And my name's Caroline Conradi. I'm a veterinary advisor in the Veterinary Exotic Notifiable Disease Unit within the Animal and Plant Health Agency. So if you suspect a notifiable animal disease, which in this case we're concerning rabies, you must report it immediately to APHA by calling the Defra Rural Services helpline. And there's a different contact telephone number for England and Wales. And in Scotland, you contact us through your local field services office. and failure to do so is an offence, and it's really important that you contact us as soon as possible if you have any suspicions, so we can take out an investigation. And reports can be made by any person at any time. So this line is manned, and we will get back to you and discuss the case. So at any time by any person. And APHA will confirm with you what exotic notified disease is suspected and why, along with determining the location and type of premises, whether that be in someone's home, a veterinary practice or a farm, the species involved and the number of those species affected, and the clinical signs that they're showing. Verbal restrictions would then be served, and the caller would be advised not to move animals, people and things on or off the premises until APHA can visit to investigate. And the concern with rabies is a zoonosis, so the relevant public health agency would also be informed, just as there was a risk to the animal and public health here. And we have two different ways in which APHA might respond to a particular case. So we have what's known as a consultation case, which is where, for example, a private vet would report a low level of suspicion of disease, and they would remain at the premises. The investigation can be conducted as a consultation case, and no formal restrictions would be served initially for these cases. An APHA field vet is in effect consulting with the private vet to see if there are grounds for suspecting that notifiable disease. And if the disease cannot be ruled out, restrictions must be served, and the investigation would then become a report case. So a report case is where a disease might be reported from a member of the public or a private vet who is not at the premises, for example, and then the investigation is conducted as a report case, and formal restrictions would then be served on those cases. So what happens when suspicion of rabies is reported? So first of all, we'd get that suspicion reported to APHA through the rural helpline using those numbers displayed before. So that could be a telephone call from an owner of a pet or a farmer or a vet who is concerned that their animal is showing suspicion of rabies signs. So the APHA duty vet would then gather information about that case, taking histories such as where the animal might have travelled from. If it was a pet, is it compliant with requirements for rabies to come into the country? Where does it live? What clinical signs is it showing? And have we got concerns that we need to go out and do a visit? So if we can't rule disease out, it's likely that we will need to come and visit to check over that animal to make sure if the animal is showing the signs that the caller thinks they're displaying. And we'd serve formal restrictions at the time of the call. And then we would carry out a veterinary investigation. And in that veterinary investigation, this is our visit to the premises where we're going to check the animal to see if it's showing the signs and are we still concerned it may have a notifiable disease, such as rabies. So we need to maintain high standards of biosecurity when we enter those premises. So we are concerned about a disease which we may not have here. We don't want to spread it, and we want to keep everything as clean as possible. So we would also serve the written restrictions, unless it was a consultation case where we would only serve those once we've decided it may be that notifiable disease. And we need to record the exact location of the affected animal using GPS. And why do we do that? Because that helps us define the site of the suspect animal and the area under restriction, which is used to set up control zones on disease confirmation. So it's really important we get that map reference of exactly where the animal is. And we need to keep the owner informed of exactly what we're doing and all actions we're taking. Particularly relevant if this is a rabies suspect in a pet in someone's home, and be mindful of the other people who might also be in that household and be affected. We'd inspect all animals and clinically examine those affected, provided it is safe to do so. Clearly, if we're worried about a very aggressive animal with concern for rabies, our safety and safety of others around us is also important, as well as looking at that animal. So we may need to restrain the animal to examine it properly and wear appropriate protective, personal protective equipment known as PPE. And we'd gather and record all the information we need and take that history of the animal and anywhere it might have been and people in contact to complete the disease report form. Contact would then be made with a central team of veterinary advisors in the Veterinary Exotic Notifiable Disease Unit, known as VENDU, to discuss the case with the field vet. So just an example of some of the equipment we might need to use. So on the left-hand side, we've got a rabies suit. So this is a thick suit. So if somebody gets bitten, it doesn't get through, penetrate that suit. It's quite alarming perhaps to see someone in that, but it's just full safety gear to make sure that that person is protected. And on the right-hand side, we've got a couple of cages. So these are secure crates for transporting a cat or a dog, which are metal with a double door system and ventilation. And then there are some cat gauntlets should you need to try and handle a cat, again worrying about the bite risk. But you may have the suit on, and an example of some graspers to try and hold an animal. And there may be some other pieces of equipment we would use as well. So once the veterinary investigation has been carried out, can we then rule out disease? So we'd have some various options depending on what we're thinking at this stage. So if we don't suspect disease, then restrictions can be lifted and there's no further action taken on that case. However, if we can't rule out disease, but perhaps the signs are only mild, so maybe it's only a mild behavioural change in a pet, but we're still concerned for rabies, then we might need to move the animal to an isolation facility to monitor it further. And that would be transported, monitored for a number of days, and then wait to see if the signs get worse or perhaps improve, and the animal might be returned. Alternatively, if we've got strong suspicion and we really can't rule out suspicion of rabies, perhaps there's been a very marked behavioural change, in that case, we sadly might need to consider euthanasia and submission of samples if rabies is really a concern here. So in those kind of scenarios where we can't rule out disease, the animal would require euthanasia for testing. We can't test the live animal. The notice would need to be served to euthanize the animal, which could be voluntary. Obviously, if this is someone's pet and has become very aggressive or has bitten a member of the family or a member of the public, they might equally be concerned and submit to voluntary euthanasia, or we may need to serve compulsory euthanasia dependent on the gravity of the situation and the nature of those presenting signs. And we might need to sedate the animal prior to euthanasia. Public health agencies would also be involved, thinking around things like biting, scratching of members of the public, which would be a worry for transmitting the disease from the animal to humans, and any people that have been in contact with this animal, if it's been out and about or within the household, who they might need to be aware and contact. The whole animal would then be transported in a suitable transport container, such as the one shown in the previous pictures, to the laboratory for testing. And those restrictions which we've put would remain in force pending testing results.
Hi, so I'm Lorraine McElhinney, and I'm the head of the National Reference Laboratory for Human and Animal Rabies at APHA. And I'm just going to give you a brief description of what happens at the lab when we have a suspect animal. So this is just an overview of the type of testing that we do at the APHA lab. So we'll test all deaths in quarantine, any animal suspects that are euthanized and sampling and testing is required, any illegal imports for which the veterinary teams decide that they need to be euthanized and sampled. And then we have some surveillance for zoo bats, passive surveillance, and also suspect bats that will come in and we will test. On top of that, we do a lot of serology in the laboratory, and this is mainly post-vaccinal. So where pets have had vaccinations, we'll sample their bloods for pet travel schemes, for example, and we can do a lot of occupational health human serology as well. And as the ref lab for human diagnostics as well as animals, we will also do ante-mortem or post-mortem diagnosis. So there is no ante-mortem diagnostic validated tests. So all of the tests that we do on a suspect animal are post-mortem. So the tests that we use, there is a number that we can use, But the one at the top there, immunofluorescent staining, this is called the fluorescent antibody test. We remove brain from the carcass. So for suspect cases, we receive the whole carcass. So we will go in, we'll remove the brain, and from the brain, we'll remove the sections of the brain, the medulla, hippocampus, and cerebellum from the brainstem area, and we'll test those using FAT. We have a virus isolation test as well that used to be routinely diagnostic, but now we tend to only use that for research purposes. There's histological staining, immunohistochemistry tests that are available to us, but again, they tend to be not so much diagnostic, but more for characterization and understanding the virus, so more for research. We have some molecular tests, and now those molecular tests are also approved by the OIE for primary diagnostics. So they are to detect viral RNA. And there's also serology, I mentioned earlier. Now, you don't tend to use serology for diagnostic results because you don't tend to see an antibody response until the very latter stages of the disease. And this is why we can use those virus neutralization tests in healthy animals to indicate that they've been vaccinated. So the fluorescent antibody test, the FAT, this is the gold standard, and we're looking for rabies antigen. So as I mentioned, we'll remove the brain, and then we'll remove the particular areas within the brain where we believe most virus and therefore antigen will collect, so around the brainstem region. We do brain impressions of those tissues onto a slide, fix it in acetone, and then we'll stain that with an antibody against the nuclear protein of the rabies virus with a fluorescent label. Where you get the fluorescence, specifically, you will see apple green staining, and we use a fluorescent microscope, as you can see in the picture there. And once we get into the lab and we've removed the brain, this test is actually quite rapid. So it's about 2 hours within the lab, but I would say between 3 and 4 hours from the sample receiving at APHA to us reporting out the results. So it's very quick, very sensitive and highly specific test. But where possible, we really do need to have fresh samples. So the other test that I mentioned was the molecular assays. And so we do FAT, but also we will use confirmatory real-time PCR assays, and they're looking for the viral RNA. So from when you have the RNA, which is extracted within our high containment labs, you can again get a very rapid result. So within 2 hours of receiving the RNA from outside, we can have that result, and the results are automatically sent through the system and can be picked up remotely by our staff. So we have 2 assays, one to detect all lyssaviruses and one that will also differentiate between different lyssaviruses. So the classical dog rabies type lyssavirus, but also the bat viruses. So we'll use our TACMAN assay to rule out any suspect cases due to EBLV1 or EBLV2, the bat rabies lyssaviruses that we have in the UK. When we have a positive result, we'll also use a classical PCR to generate some template, and we'll send that off for sequence analysis. And this is to help us to try to characterize the virus and understand the source. We'll also do some whole genome sequencing. And so to help us, again, look fully at the virus and any subsequent samples that arise if the outbreak continues and expands. Rabies serology, as I mentioned, we would only really use this very rarely in late disease, and only if we knew that the animal had not been vaccinated. So the assays that I've mentioned here, the virus neutralization assay or the ELISA, cannot differentiate between rabies disease or vaccination. And so we need to, when we take a blood sample for post-vaccinal, that animal needs to be healthy. The virus neutralization test, because it involves live virus, has to be performed in the high containment use and takes a few days to get a result. The ELISA, on the other hand, can be undertaken outside of high containment, but unfortunately it's not accepted for pet travel at the moment. However, we're using it for this emergency response to confirm whether the animal has been vaccinated or not. But the ELISA results cannot be used for onward travel. The assay is very good and is actually used for fox and human serology widely. So we have full confidence in the results that are being used to facilitate pet movements from quarantine to home isolation in these exceptional circumstances. Both ELISA tests, the ELISA and the FAVN, can be adapted for high throughput serology. All of the testing that we have at APHA is accredited to ISO 17025. So this last one just gives some usual turnaround time. So as I mentioned, our diagnosticians are available. They're on call 24/7. The carcasses will arrive at security and our on-call staff notified, and they will go into the high containment lab and start processing that FAT. And in most cases, we'll report out the results within 4 hours of receipt at APHA. The molecular tests take a little longer, and most of them are reported within 24 hours, but actually in practice, we will get those within a few hours of the FAT results. Sequencing and characterization takes a little longer, so 48 hours. All of the results that we get from the lab are reported directly to Vendo and then disseminated through the NDI reports. What positive cases we'll also phone through just to make sure that there's rapid communication of any positive results. Unfortunately, the carcass cannot be returned once it's gone into the high containment lab. The ELISA serology that's currently being used at the moment, we say that in general, the results are going out within 36 hours of receipt. But if samples arrive before 10 o'clock in the morning, then we're actually reporting those out later on in the afternoon. And border staff can check the results via an online tracker that we've set up. So thank you very much.
Hello, everyone. I am Tarindu Sri Premachandra. I am a policy advisor for rabies in exotic disease control policy team in Defra. In this presentation, I will talk about rabies outbreak response and control measures, which we will use to control a rabies outbreak in Great Britain. So the UK is classical rabies free since the beginning of the 20th century. The last recorded case of classical rabies in terrestrial mammals were in 1922. This happened when servicemen returning from war smuggled rabid dogs back to Britain. The disease was controlled and eradicated in terrestrial animals at that point. After that, there have been 2 times where imported dogs had rabies. This was in 1969 and 2008. These incidents did not affect the UK classical rabies-free status because these puppies were kept under quarantine conditions. But a licensed bat handler died in Scotland in 2002 because of European bat lice virus, which is also commonly known as bat rabies. I think it is a good thing to note that currently we do get about 2 or 3 bat rabies cases annually. But again, these do not affect the UK rabies-free status under international guidelines. There's a segment on wildlife rabies in this webinar, and you can find more information on bat rabies there. Rabies is a notifiable disease, so it is a legal requirement to report suspicion of rabies to Animal and Plant Health Agency. If there's an outbreak, we have various legislative powers which we can use to control it. These are primarily set out in the rabies control order, which can be used for declaring infected places and zones. Then there are other legislative powers to support the rabies control order. There is rabies importation of dogs, cats, and other mammals order, which mainly controls the importing of any animals belonging to a specific list of mammals. Then the rabies compensation order governs the amount that payable in respect of animal euthanized under the rabies control order. The Animal Health Act enables local authorities and police to act. And the movement of animal restriction order allows a veterinary inspector to impose restrictions on requirements to prevent the spread of the disease. And finally, there is a zoonosis order, which provides powers to enter a premises and take an animal for the purpose of testing. The rabies control order is not underpinned by EU legislation, so it offers a lot of flexibility when compared to legislations of other animal diseases. It allows us to declare infected places and zones after a veterinary investigation. However, unlike the other diseases such as avian influenza or blue tongue control, we don't have to set the size of the zones. They can be any size and shapes and can be divided into subzones, which could have different measures within these subzones. Then we can apply disease control measures within these declared zones. This be movement and behavioral restrictions, applying muzzles on dogs in public places, giving compulsory vaccination to domestic animals, or it be allowing to cull an animal infected by rabies. Some of these disease control measures, such as movement restrictions, can be allowed under licenses, depending on the risks as well. On this table, this table shows the possible scenarios and control measures involving the disease spreading to wildlife. We are potentially an infected fox and a domestic dog, a domestic non-imported dog. This is an example, and it does not cover all possible scenarios, but it is one of the most likely ones. More detailed version of this table can be found in rabies control strategy. Application of all or some of these control measures will be determined by various factors. For example, depending on the severity of the incident and the likely spread of the disease. The overall rabies outbreak control process is a massive multi-agency response which connects national and local level structures. Each of these organizations have to work together to control an outbreak as soon as possible. As you can see in this slide, Defra, APHA, other government departments, they all have to get together to address the situation as soon as possible. In terms of dealing with an outbreak, the way forward depends on the circumstances surrounding the outbreak, but it will start with a discussion with all the structures that are involved in the response. This include APHA, Devolved Administration, COMS, field operations, local authorities, and operational partners such as Royal Army Veterinary Corps and the National Police Chief Council. Public health collaboration and response is also very important, as rabies is a very serious zoonotic disease. Each of these agencies have set roles and responsibilities to manage the outbreak. Defra leads the animal disease outbreak control response in England by establishing a number of structures to coordinate and support the disease control response. The strategic decisions and measures are based on advice from experts, veterinarians, policy, economists, operational partners, and stakeholders. When needed, key GB-wide policies will be submitted to the Animal Disease Policy Group for approval. This is also and then there is also the Rabies Core Group. They also advise and provide recommendations to develop policy solution. The delivery of local and national operational functions is done via executive agencies such as APHA. Devolved administrations would lead the outbreak control in their respective administrations, and they have similar plans within their areas. Then the rabies control strategy sets out the framework detailing how a rabies outbreak would be managed in Great Britain. This strategy provides a general control principle for the most likely scenarios for cases of classical rabies. This strategy does not provide operational advice on how to deal with an outbreak. This is set down in the contingency plan for exotic notifiable disease of animals. Rabies control, the exit strategy. Once the outbreak is controlled, drawing up an exit strategy would largely be influenced by the circumstances. So this could be things like size of the infection, affected animal species, and number of contact animals. A key factor in determining an exit strategy for the lifting of restrictions would be the source of infection. National Emergency Epidemiology Group advise relevant CVOs and policy leads on this, and then they take forward actions based on that advice. It would take up to 2 years for us to regain our OIE disease-free status for rabies following an outbreak. So when a rabies case has been detected in wildlife, we would apply for disease-free status to be reinstated 2 years after the last detected case. During this time, we'll do passive surveillance and testing for fox carcasses to make sure no more new cases are detected. I think this concludes my presentation on rabies outbreak response, and I hope you will find it useful. I will leave you with some useful links for further information. Thank you.
Hello, my name is Dr. Catherine Russell and I'm Head of Emerging Infections and Zoonoses at the UK Health Security Agency. Today I'm just going to talk a little bit about the public health aspects of rabies and post-exposure treatment. As you've heard already, rabies is an important acute viral encephalomyelitis caused by infection with classic rabies virus or other related lyssaviruses. The domestic dog is the major reservoir and responsible for over 99% of human deaths globally. As you've already heard in this session, it's spread through the saliva of an infected animal. And although effective post-exposure treatment is available, once symptoms of rabies start, it's almost entirely fatal. There are up to 70,000 deaths a year globally, and sadly the majority of these deaths occur in children, who are more likely to sustain severe head and neck bites. And controlling rabies in animals is really the mainstay for preventing human infections. So human rabies infections present in two main ways, but often the first symptoms that people report are pain or tingling or numbness at the site of the bite. This then radiates centrally, and as with animals, it can present in either a furious or a paralytic form. These may present slightly differently in terms of clinical manifestations, but unfortunately the outcome is generally the same. Since 1902, there have been 29 human rabies cases in the UK, the majority of which were linked to dog bites in the Indian subcontinent. Unfortunately, the commonalities are that all except the most recent case hadn't received any post-exposure treatment before they presented with symptoms. In the most recent case, the individual did receive post-exposure treatment, but this was quite delayed after the bite had occurred. So post-exposure treatment is made up of two main parts. Wound care immediately after a bite, scratch or other exposure is vital to remove as much virus from the site as possible. Then a risk assessment of the exposure should be carried out. The UKHSA RIGS or Rabies and Immunoglobulin Service team can be contacted by health professionals to help carry out the risk assessment and provide rabies vaccine and immunoglobulin. The arrangements may be slightly different in our devolved administrations, but their public health agencies will also be able to provide advice on where to access risk assessments and post-exposure treatment. So this post-exposure treatment, as I've mentioned, involves rabies vaccine and human rabies immunoglobulin. The vaccine is typically given on over 4 doses on days 0, 3, 7 and 21. And we try and give the rabies immunoglobulin as early as possible after that exposure. Now, vaccine is the mainstay, and I'll show you a graph in a moment to explain this. But when we give immunoglobulin, we try and give it at the wound site to mop up that live virus and give that rapid protection. And it's not as useful if people have already started and been well established on their vaccine course. This is a busy chart, but it shows some key bits of information about our post-exposure treatment that we offer. If you focus on the red line, this shows the development of vaccine-induced antibody responses. So you can see it takes a little bit of time for a vaccine response to be started, and it's really important to get that vaccine started as soon as possible after an exposure. The other line to look at is the purple one, which shows passive immunity provided by rabies immunoglobulin. This is given in the period straight after a bite or other exposure to provide antibody before the person starts to make their own antibody from their vaccine course. When we're carrying out a risk assessment, we need a number of different pieces of information to ensure we're conducting a thorough assessment with all the information available. So we need to know the demographics of the individual, when the exposure occurred, where and what type of animal was involved, and what exactly the exposure involved and where it occurred on the body. We also need details of the past medical history. Is the individual immunosuppressed, or have they had any other treatment already? We use a combination of the risks from the animal, the country involved, along with details of the bite or scratch to come up with what we call a composite rabies risk score. Then we assess whether the patient has had any previous rabies vaccine to determine what treatment they need next. An example might be a person who was bitten badly on the leg in India by a dog. They would be categorised as having a high risk category 3 exposure. And if they've never received any rabies vaccine in the past, they'll need a course of 4 doses of rabies vaccine and immunoglobulin as part of their post-exposure treatment. What's really important to note here is even those who have had a pre-exposure course of vaccine still require 2 doses of vaccine after a bite or scratch or other exposure. Therefore, it's really important to ensure that you always report any exposures and ensure a risk assessment is carried out as soon as possible afterwards. This chart shows the trends in calls we've received in England over the past 10 years. As you'll see from the grey line at the top of the graph, calls about exposures to dogs overseas form the majority of our work. But you'll note there's a drop-off significantly in 2020 during the pandemic when international travel decreased. The other spike you'll see towards the end of 2018 in the dark orange line is calls related to cat exposures. And sadly, that peak we saw then was related to the last human case we had in the UK, which was a cat exposure from Morocco. You'll also see the blue line peaks every year in the summer months, and this relates to the regular seasonal trend we see in bat exposures here in the UK. So you'll be aware already of much of this information, but if an animal is suspected of being illegally imported, it needs to be referred to trading standards immediately. An illegally imported animal is not necessarily the same as an animal with suspected rabies, although it's possible for an illegally imported animal to present with concerning symptoms and rabies to be part of the differential diagnosis. Wherever there is a suspicion of rabies in an animal, public health authorities will be guided on the risk in the animal by APHA or Defra, but the decision to offer post-exposure treatment to people involved is always made by UKHSA or another public health agency, and not the vet or trading standards, as a full medical risk assessment of the patient is required. If an animal is under observation or in quarantine, there is not necessarily a need to start post-exposure treatment straight away, unless there is a high suspicion of rabies in the animal. There's usually time to await the outcome of testing. However, UKHSA should always be informed if there's a suspicion of rabies and will advise on the need for post-exposure treatment. This slide just shows some links for other information about human aspects of rabies, along with 2 rabies leaflets we've developed for travellers and also for contact with bats. Thank you very much.
Thank you very much, everybody, for listening to this series of webinars, and I hope you have found them useful. For further information, we would just like to emphasise that please remember rabies is a notifiable animal disease. It can be prevented by vaccinating animals and by post-exposure treatment of people. So it's really important that if you suspect it in an animal, you must report it immediately. Please contact the following. In England, the Defra Rural Services Helpline. In Scotland, it will depend upon the different region that you're present in, but the numbers are here on the screen for those different areas. And in Wales, again, it's a separate number to that for England and Scotland. For more information, please see the Defra website. As you see here, rabies, how to spot and report disease in animals. You'll be able to Google that quite easily. And if you need more information, please do contact the Exotic Disease Policy team at exotic.disease.policy@defra.gov.uk, and we will be able to help you with any of your concerns. Thank you.